Okay, thank you. All right, so good morning. So this week, we are technically we're kind of beginning week five in the semester, but the first week was kind of an intro week, so it's kind of like our fourth week of doing um, study. So ideally, again, you guys do about 100 points a week. So right now, you should ideally be at around 300 points or so, I would say. So here's the week we're starting right here, which is technically, like I say, unit five. But anyway, we're going to be doing muscle physiology today. I saw, I don't know, maybe seven or so, about a little over around half of you have done this case study. But anyway, I want to talk about how a muscle contracts, basically. All right, so that goes along with the case study. So that's kind of where you should be around that 300, 400 point mark. Um, Midterm grades are due here in a couple of weeks. And we're required to give an actual letter grade, not just a pass fail. So, which is tricky in a class like this because you work at your own pace. So, so when you're at midterm, you should be at halfway, ideally, which would be about 600 points. So, but I mean, some students are at 100 points, 200 points. That's fine if that's the pace they're at. So, anyway, don't put much stock in midterm grades, is my point. You'll probably have an A regardless. I think I pretty much give A's to everybody, unless you're at like almost no points, which I don't think anybody's pretty close to that. All right. So anyway, I wanted to mention that.
Kevin, do you do a lot of labs in this, what, a lot in this class?
Yeah, this semester, every semester, yeah.
Oh, okay.
Yeah. Usually 4 to 6, probably. Yeah. So let's see. So before we get into the case study, there are a couple things we need to talk about regarding muscle physiology. I know that we've talked about skeletal a little bit this semester, not a heck of a lot. There's not much in the way of WIDS doesn't require they just don't require that. So anyway, so yeah, we just don't do much with skeletal physiology. So here we go. Here we go. So we don't do a heck of a lot with skeletal physiology. We talk a little bit about it in right general anatomy. We talked about like fracture healing, and I know I talked about Wolff's Law a little bit. Stress on bone builds bone, right? Osteoblast, osteoclast activity. You guys remember those terms from general? So basically, bone physiology involves a lot of calcium, right? So if we need to store calcium, it goes to the skeletal system. Okay. If the body needs calcium, so not the skeletal system, but other cells, we can go to the skeletal system and get calcium. So there's this kind of like storage unit or kind of like a perpetual gas tank of calcium available, which is good because this is what uses the calcium. Okay, so when we get to muscles and muscle physiology, calcium is one of the major players. So, real quickly, because again, I don't want to spend too much time on the anatomy, but I do want to give you a quick little kind of background, I suppose, of a couple of things. Yeah, right. So, when we look at structural aspects of muscle, you want to, I mean, I don't know how deep in you guys got or what you remember, but you have this belly of the muscle, which we can see with the naked eye. But as we get deeper and deeper and deeper into the muscle, we could see that it's really just a bunch of bundles of tubes. Okay? And so all these bundles of tubes have different names. So these are going to be fascicles. Okay? So these bundles are fascicles. And then the fascicles are going to be bundles of fibers. So at this point, this is technically the cell. A muscle fiber is the muscle cell. Okay. And we know from cell anatomy that from organ to organ, tissue to tissue, cells are different. They have different appearances. Some are going to have thicker membranes. Some are going to have one nucleus. Some are going to have more than one nucleus. Some are going to have a few mitochondria. Some are going to have a lot of mitochondria. Okay, so we have a lot of endoplasmic reticulum, the tubes where we're going to do fat or protein synthesis. We have a lot of different things going on depending on what the organ is. So when we get to muscle, the specific cell of a muscle is called a muscle fiber. Okay, so you've probably heard the term muscle fiber at some point, and really we're talking about the cell of the muscle. So so again, the cell is always kind of the smallest functional unit, right? The smallest functional unit of organ and tissue is going to be the cellular level. When we look at muscle, we see this kind of outer layer. This is called the sarcolemma. We call it the cell membrane, right? In muscle, they call it the sarcolemma. We see this sarco coming up. Sarco means fleshy or muscle-like. So sarco or flesh. So the lemma is the membrane. So then once we get into the membrane or past the membrane and actually into the muscle cell itself, we see the sarcoplasmic reticulum, which is the same thing as endoplasmic reticulum. They just are more specific with what they label it in here. This is where calcium is also stored. So it's important to know that calcium storage is not just in bone, but it's also going to be in the muscle cells, specifically sarcoplasmic reticulum. So generally speaking, when a muscle is at rest, calcium is going to be at high levels in this sarcoplasmic reticulum. Okay, so when the muscle's at rest, calcium's kind of hanging out in here in the sarcoplasmic reticulum. As soon as an impulse comes down from a nerve, ultimately we're going to have to get calcium, not all of it. We just need some of it to leave the sarcoplasmic reticulum. So some calcium is going to leave the sarcoplasmic reticulum. It's actually going to go deeper into the muscle cell. Okay, it's going to go deeper in the fiber. Inside of the muscle cell, not only do we have the sarcoplasmic reticulum, we also have a whole bunch of filaments and fibers. Basically, kind of remember we talked when we look at the cell structures, one was cytoskeleton. So cells have kind of their own skeleton sometimes, and it's usually made up of tubes, tubules, filaments, fibers, some sort of structural feature will give a cell kind of its skeletal aspect. So in muscles, we see a whole bunch of filaments. Filaments. These are going to make up kind of the bulk of the muscle cell. So calcium is going to be stored in here. As soon as we need a contraction, that calcium is going to leave this sarcoplasmic reticulum and it's going to come down into the fibrils. And ultimately, we're going to see it attached to these filaments. That creates a contraction. So when a muscle contracts, calcium, not all of it, some calcium is going to leave. It depends on the strength of contraction that's required. If I'm just picking up, my cell phone, it doesn't weigh a heck of a lot, I don't need to recruit a lot of muscle. Okay. Whereas if I'm going to, lift up this board, or whatever, this table, I have to recruit more muscle, right? More fibers. So I might dump or require more calcium out of here. So the point is, if you're contracting a muscle, you need calcium, and it's going to dump out of this sarcoplasmic reticulum. Now, as soon as I need to relax a muscle, right, calcium's going to go back into this area, now into the sarcoplasmic reticulum. So as soon as there's no more transmission of electricity coming in, there's no more stimulus saying we need to contract it. Now we need to relax it. As soon as that comes in, everything kind of goes back to where it was. So calcium goes back into the sarcoplasmic reticulum. We'll see sodium as a player in this, and we'll look at another neurotransmitter. But the point is, calcium is going to be kind of hanging out outside of here during a muscle contraction. As soon as we need to relax, we got to get calcium back in. Now, there's still more calcium in here than out here. So we do have to pump it against its concentration gradient. So that does require ATP as well. Yeah, Ricky.
So when it stops, like when there's like a lack of calcium, like coming back, what was it? You said coming into the cell, and then that's where we get the output.
Yeah, calcium's always inside of the cell. It's
Oh,
stored in
out, right?
yeah, stored inside of the basically the endoplasmic reticulum.
Okay.
It's stored in there, so it's inside the cell, it's just stored in these tubes. And deeper into the cell, once we get into this level, that's where the calcium's going to be active.
Oh,
So it's stored in here, active over here. As soon as we don't need any more activity, it has to go back into the tubes. It's still in the cell. It's just inside of the tubes in the cell.
so if we were like deficient in calcium, like what would like that be labeled as, like as if there were a disease or something like that?
Yeah, we do have like hypocalcemia, they call it. So not enough calcium in the blood. Now, there are two, we saw it week, your second case study talked about the thyroid, but it mainly talked about metabolic aspects. So we talked about T3 and T4. But I wanted you to discuss, not discuss, I wanted you to mention, and all of you did, we did together too, that calcitonin is a third hormone from the thyroid. So calcitonin deals with calcium homeostasis. So basically, calcitonin means tones the calcium. So when you have calcium, when you have excess calcium, so hypercalcemia, so that just means you have a lot of calcium in the blood, then you're going to get calcitonin released and it's going to come into the bloodstream and say, hey, we're going to go ahead and store this into the bone. Typically, he's going to put it in the bone. Some of it will go to muscle, of course. And then at some point, though, we're now deficient in calcium, right? Because at some point, there's no calcium in the blood, which are limited amounts. So now I'm at a hypocalcemia state. And again, this is constant. We're always hyper or hypo. Now, do we stay at hyper or do we stay at hypo? That's when diseases and disorders and problems come into play. So there's a secondary hormone that comes from the parathyroid glands. So remember, the thyroid has four tiny glands in the posterior aspect called the parathyroid hormones or parathyroid glands, and they produce parathyroid hormone. Parathyroid hormone does the opposite of calcitonin. Because again, in the body, everything comes really with like antagonists, like opposites. So it's like yin and yang, up and down, left and right. So if I have a chemical that's going to pull calcium out of the blood, I'm gonna have an opposite chemical that's going to put calcium into the blood. See? So calcitonin pulls calcium out of the blood and puts it into the bone. Parathyroid hormone comes in when I'm low in calcium and says, hey, bones, we need osteoclasts to start chiseling some of the bone away to dump calcium into the blood. So there's rarely a situation, well, not rare, because our bones are so dense with calcium, it's doesn't happen that often where a patient is completely out of calcium. You're going to get it from that patient's bones might become porous, but they may be lacking those hormones. So just because you're eating calcium doesn't mean you're necessarily going to absorb it and store it. Because you need hormones, right? So if we do have a hypocalcemia situation, remember emia is a blood condition, calc just means calcium, hypo not enough. So hypocalcemia situation, we're definitely looking at the thyroid and parathyroid. We're looking at hormones for sure. And because like I say, the bones are full of calcium. They could have a severe case of osteoporosis where they're just losing bone density and calcium's being leached, from somewhere. Um, so like elderly folks generally are losing muscle mass, they're losing some bone density. Well, they still need what's the main muscle that needs to function? Well, the heart, right? So the heart needs calcium all the time. So the calcium's probably the most if we were to hierarchy the elements, it's hard to do that. But if we were to say, the number one element in the body that is necessary is probably calcium. I mean, really, truly. And the other thing about calcium, so I mean, it makes your heart contract. If you don't have calcium or you have low levels, then you're going to have heart problems. If you have heart problems, then everything goes south, right? And in America, really probably globally at this point, heart disease is the number one killer still. So there's obviously a calcium problem somewhere in there too. But part of calcium attaining and then absorbing and storing involves two other elements, or not elements, two other items. One is vitamin D. So you need vitamin D to go with calcium to get it to absorb into the guts. So if you take calcium, but you don't have the right amounts of vitamin D, then it won't absorb in the intestines. So you can't assimilate it. So you'll eat calcium and then you'll just poop it out. You're not going to take it on. So you need vitamin D, but then you also need ultraviolet rays. You need UV exposure. UV exposure will trigger vitamin D to be more active, which then triggers more absorption of calcium. There are a few other elements involved there, but and hormones. One's called cholecalciferol, and coming from the liver and the guts, the small intestines. So, you know, and calcium is a mineral, so it comes out of the dirt. So, you know, when we look at soil quality, we're looking at mineral content. So, what's the calcium levels? What are the magnesium levels? And nowadays, because of erosion and just different types of farming, it's more difficult to retain some of these elements, these minerals in the soil that we need to. So whatever's growing out of the ground isn't the vegetables and the fruits, they don't have as much mineral as maybe they once did. And so we have to fortify stuff, right? So when you read about so this is part of your nutrition class stuff. When you're talking about fortifying foods, we have to add stuff in because it doesn't have enough of it. So calcium is a big one that we need to fortify things with. But anyway, animals that eat the foods that come out of the ground, they're also then not getting enough of those minerals. Then we eat the meat from that animal, So you get the idea. We're just more mineral deficient maybe than we have been in a while. So it's important. So calcium deficiencies are a big one. And again, it comes back then to the thyroid gland, because the thyroid and parathyroid regulate kind of what we call calcium homeostasis. So basically in the body, in the clinical setting, calcium homeostasis and glucose homeostasis. Those are the two things we're looking at big time, right? Are they diabetic or not? Basically, that's the glucose homeostasis. And why is that important? Because all of these cells in here have a lot of mitochondria, especially muscle, tons of mitochondria, and the mitochondria, back it up, see all of them. These mitochondria use the glucose to get ATP going, and you need ATP to get calcium pumped back in here. Because remember, from last week, we learned you need ATP if you're pumping against a concentration gradient, or if you have a vesicle that's delivering stuff, so a vehicle. Okay, so you need ATP. And you get ATP from glucose and oxygen. So you have to breathe and you have to get glucose. That's why, again, glucose homeostasis is a big deal in the clinic. Because if a patient comes in and they're lacking energy, they're sluggish, they're sleeping all the time, that's a problem. Yeah, we're not bears. We don't sleep. We'd like to probably, but yeah, I mean, we don't sleep days and days and days and weeks and months on end. And you have patients that come in, especially older patients that, they may be carrying a chronic UTI. They may be, who knows? But either way, their energy levels are low. They're not get so we look at glucose homeostasis, and they might be type 2 diet. They might be diabetic. They might not be eating enough. They might be eating cat food. We don't know. So in the clinical setting, this is what we're looking at. They might be gaunt, frail, lacking energy. So yeah, the homeostasis is all wacky. And part of it's at this level. Then they have heart problems, right? Because if they're not eating right, if they're not getting any energy, activity, and the proper elements to keep everything rolling at a normal pace, they wake up dead, heart attacks or heart failure or whatever. All right. So anyhow, this is a muscle cell. So as we look, we go back here, we basically are blowing this up onto the next slide. So this is kind of where all the action happens once we get the muscle to contract. We're going to have to dump the calcium out of here deeper into the muscle to these myofilaments. So the myofilaments, you can kind of see they represent them as being kind of thick and thin. So we're going to look at that in a few minutes. We're going to see thick filaments and thin filaments. They're going to be kind of sliding and gliding together over one another, and that's causing the muscle to contract. And again, it's the presence of calcium that's going to basically expose what we call the binding site. So that's what I want to talk about now for just a few minutes. There. These are what those filaments look like, and they're thick and thin. You can see these dark, they're not dark, but the blue ones are the thick ones, and these red ones are thin. And the thin ones are called actin. Okay, you can see right here, actin, actin. Okay, so this is an actin muscle filament. And actin is basically, as you can see, it's kind of like two beaded kind of pearl necklaces kind of twisted. So I call it like a double helix. So it's just basically two long beads spun together. Now, again, when we're looking at structures in the body, they're pretty much always going to be protein. So again, it ends in the letters IN. IN. That's a protein ends in IN. So that's a good indicator that it's a protein. Okay. And proteins are the strands or globules. So you can see it says strands. So these are protein strands with a bunch of amino acids strung together. So what we're going to see with actin, so actin is considered the thin filament. Okay? Actin is thin. Myosin is the thick filament. Okay? So actin and myosin, thin and thick. Now, actin has what we call the myosin binding site. So myosin is going to bind to actin on these, what they call binding sites, active sites or binding sites. Now, when a muscle is relaxed, the binding sites are covered up. So myosin cannot bind to actin when these binding sites are covered up. So you can see on this illustration, these little kind of divots in here, those are the binding sites, and you can see how they're representing them being covered up. Okay, so you can see how they're covered up. Now, they're covered up with something called tropomyosin. So this is another strand of protein kind of intertwined in that double helix called tropomyosin. Tropo means change. Tropomyosin. So tropomyosin is an extra protein that's going to be, it's another extra filament that's basically covering up the binding sites. Okay. Now here's where it gets kind of cool. This, you can see these little yellow blobs. These are called troponin complexes. I don't have the word complex written up there, but sometimes you'll see it as troponin complex. Okay, you can see these are the troponin complexes. And these are, again, another protein that ends in IN. These little structural proteins are attached to the tropomyosin. You guys see that? How these guys are attached there? So what happens to get a, when we need a muscle to contract, we have to expose all of these little divots, all these binding sites, so these heads can kind of fit right into there. Okay? So how do I get this tropomyosin out of the way? So we need to get tropomyosin out of the way. So what we do is we're going to add calcium to this troponin complex. So as soon as I dump calcium into the muscle fiber, the muscle cell, so I have to get it out of the sarcoplasmic reticulum. It's going to come over here and attach to these troponin complexes. As soon as calcium attaches to those, it causes them to make a little move. So they twist a little bit. They just kind of doop. And as they do that, it pulls this troponin or the tropomyosin out of the way. So as soon as I drop calcium onto here, it causes a little bit of a shift. And what it does is it exposes the binding site. Then once I remove calcium, this little bugger is going to go back to where it was, going to switch back, causing all of these to cover up the binding sites. Okay. So calcium triggers that troponin complex to move, which then causes the tropomyosin to expose all those binding sites. There's a lot of action going on the micro scale. They call this the sliding filament model. Sliding filament model. It used to be called the sliding filament theory. You know, before all the big microscopes, they kind of theorized this is what was going on. And I don't know, at some point they got it all figured out. All right. So the myosin heads attached to the actin. Going back out to the muscle cell. Now, in order to get the muscle so we kind of went all the way in. We went to the deepest part of the muscle, the filament level, expose the binding site. We get attachment of the myosin heads to the actin, and we get movement. How do we get calcium out of the sarcoplasmic reticulum? How does calcium know when it needs to leave the sarcoplasmic reticulum and go deeper into how does calcium know it's time to leave here, come out here, and kind of come down into these deeper levels? How does it know to leave here and come in here? It's going to get triggered by a domino, and it's going to get triggered by sodium. Okay? Sodium, remember, lives outside of a cell. We learned last week, generally speaking, sodium likes to be in high levels outside of the cell. Sodium kind of lives out here. Some sodium's in here, but remember, we have to kind of keep it pumping it out because we don't want, generally speaking, we like sodium out here. Otherwise, water chases it and we end up getting the cell gets all bloated up. Okay, so we have sodium out here. As soon as we need so it's kind of lingering around in here. As soon as we need a muscle to contract, though, we're going to get an influx of sodium. So we're going to get some changes with sodium. The sodium domino is going to fall, basically creating this kind of ripple effect that's going to tell calcium it needs to leave the sarcoplasmic reticulum. So basically, we're going to see three dominoes. The first domino, the fall, is going to be up here. Okay. So when we look at this, we see this is a neuron coming down. Specifically, this would be a motor neuron. This again, any neurons are either motor or sensory. Okay, if it's a motor neuron, it's plugging into muscle. So where that motor neuron ends, we call this an axon terminal. Call it a presynaptic junction or a presynaptic cleft. Okay. So we're going to see the words presynapse, axon terminal, motor end plate, a lot of different terms that have to do with the nerve and the muscle coming together. So where this nerve plugs in, we can see a couple little vesicles that contain a new chemical. We haven't seen this chemical much yet, but here's the abbreviation, ACH. ACH stands for acetylcholine. Okay, acetylcholine is Acetylcholine that's a neurotransmitter, right? It's in the nerve ending, it's in the bell of the nerve, the synaptic knob. This is a neurotransmitter. And that acetylcholine is the primary neurotransmitter to get a muscle to contract. Right? So basically, what happens is a nerve stimulus comes down here, triggers acetylcholine to get released from the synaptic knob, okay, from this axon terminal. Remember, this is an axon. Axon brings impulses away from the cell body. So this is an axon. At the end of the axon are these synaptic vesicles that contain acetylcholine. Soon as a stimulus comes down to trigger acetylcholine to leave, it's going to travel out of this synaptic knob and it's going to go across this synaptic cleft, it's called, to what they call a postsynapse, which is going to be the muscle. And on this muscle, this is the sarcolemma. Remember, this is the muscle, the cell wall. We're going to have acetylcholine receptor sites. So that's what these little guys are here. These are receptor site. Well, this is a receptor site, and this is a filled receptor site with acetylcholine. Okay, so ping, we hit that. Acetylcholine gets dumped into this synaptic cleft and then gets picked up by these receptor sites. So these acetylcholine receptor sites pick up that acetylcholine. That is going to trigger, so that's your first domino. Acetylcholine, boom, falls, hits the next domino, which is sodium. So then sodium levels are going to be very high inside of the cell now. So then sodium's going to go and it's going to basically wake up that calcium that's stored in the sarcoplasmic reticulum and tell that you need to get out of there and attach to the troponin complex. So acetylcholine, then sodium, then calcium. Okay. And then what happens? Muscle needs to relax, so there's no more stimulus coming down here. So no stimulus. So then we have a chemical in here called ACHE, acetylcholine esterase. It's got that ASE on the end. It's like ache. I'm making a capital E. But anyway, ACHE is acetylcholine esterase. Remember, ASE is an enzyme, so it's going to break things down. So when acetylcholine's in here and we no longer need a muscle to contract, the enzyme comes in and basically kicks it out of here. It kind of breaks it down, sends it back. So then I've got acetylcholine back in here. So it basically set the dominoes back up. Okay. So again, the dominoes, we've got them set up. We've got the ACH domino, and we've got the sodium domino, and then we've got the calcium domino. To get a muscle contract, you just have to tap that first one, and that's going to fall into that one, boom, 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 and then you get the muscle contract. In order to relax it, we have to set them back up. So calcium gets set up, sodium set up, acetylcholine gets set up. Metaphorically. So acetylcholinesterase, the enzyme in here, is going to tell acetylcholine you're done. You need to go back to the synaptic knob. So then the muscle relaxes. So in our case study, we're going to see two different patients, one of which doesn't have enough and one has too much. We're going to see a hyper and a hypo. So if I don't have any acetylcholinesterase, so if I fill these up, I'm going to get a muscle to contract, right? If these never go back home, then the muscle is going to stay contracted. Right? That's a problem, right? We don't want the heart or any of our other muscles to stay contracted. We need them to relax. Yeah. And then I have another issue where acetylcholinesterase is coming in and it's basic, or there's maybe some sort of antibody that destroys acetylcholinesterase, kind of one of your own antibodies. So you never ever really get. So you're constantly getting maybe getting calcium, or I mean acetylcholine is getting eaten up all the time. So it triggers acetylcholinesterase to be hyperactive. So we have a situation where it would be the opposite. So now acetylcholinesterase is basically eating away the acetylcholine before it even does its job. So then there's no muscle contraction. So we'll see hyper and hypo. So let's take a look at that. Go into content. Case study. This case study is straightforward. There are, my point is, it's not going to take us too terribly long to get through it. Um, so this case study has some terminology at the front end. So basically, a lot of the terms that we went through, some of the anatomy, some of the physiological stuff is going to be in here. So and all you'll need to do is just put the letter. You don't need to type out, just put the letter in here. Okay, so thin contractile protein. Okay, remember, actin is the thin filament. And somewhere there's the thick one, thick filament. Okay, so that's going to be the myosin, for instance. Neurotransmitter from choline, that's going to be acetylcholine, and it's et cetera. All right. And then at the bottom, after number 16, there's a little spot there. It says, using the sliding filament theory, explain or draw the process of a sarcomere shortening. Basically, I just want you to be able to discuss kind of what I went through. Acetylcholine gets released from the synaptic knob, triggering sodium to rush into the cell, which then triggers calcium to leave the sarcoplasmic reticulum, which then attaches to the troponin complex, moving tropomyosin out of the way, exposing the myosin binding site. So that's what I'm looking for that. And by the way, you need ATP to detach that connection. So once we get the cross bridges, so when the myosin head joins up with the actin, it's going to, in order to release that, so even if I dump, so if I say, all right, no more calcium, right? So they're still going to stay attached. But we're just not going to get them detached and reattached. So what I need is ATP to detach it. So ATP causes the detaching, in essence, of the actin and myosin. So you need ATP to relax the muscle. All right, so let's look at, that's the first part of it. The rest of this case study involves two separate patients. One comes in, he works at the CDC, he's working with a neurotoxin called sarin. And while he's working, something happens and he starts to get dizzy, heart pounding harder than ever, shortness of breath, sluggish, etc. So it starts cramping up, falls down, hits his head, and is unconscious. So they take him to the clinic. So again, first off, what symptoms is Jeff experiencing? Well, again, we just saw it up here. We put, he's dizzy. He's got heart tachycardia, basically, SOB or shortness of breath. Muscles are cramping. Yeah. So those are the main symptoms, I'd say, for Jeff. And then what's the role of ACHE? Again, ACHE is going to have a lot to do with it's the enzyme responsible for getting acetylcholine out of that synaptic area. Okay, so acetylcholinesterase is an enzyme at the neuromuscular junction that kicks acetylcholine back to the synaptic knob or back to the presynapse. So basically, it's going to trigger muscles to relax. Yeah. Rick.
So can I just think of acetylcholinesterase as like an eraser? So
Yeah, it's kind of like an eraser. Yeah, it erases what's going on. So then we can start over with another contraction. Yep. Perfect. So a lot of enzymes kind of work that way. They just kind of get the reaction to speed up and be done with it. Yep. Yeah. So then we admit Jeff. We want to know what his deal is. Pretty much as we look at his numbers, they all look pretty good, except for this one, his ACHE activity test. So it should be at 100. all the time. Should have 100 on there. He's only at a 44. Okay, so which of Jeff's levels are abnormal? Everything else looks pretty good. Which of Jeff's levels are abnormal? Definitely that ACHE activity test. He's only at 44. So, what's the mechanism of action of sarin or sarin-like chemical? So, you'd have to Google that, of course, but you could probably, if you did Google it, you'd probably find that it affects acetylcholinesterase activity, since that's what Jeff was exposed to, and that's the number that's low. So how would exposure affect amounts of sodium going into the muscle cell? Okay, so affect sodium going into the muscle cell. So remember, he's got, so this domino fell and it knocked sodium. Well, we need to see the cholinesterase to set everything back up. So we don't have that. He's lacking. He's got minimal amount. So where's the sodium domino? It's fallen. That means sodium is in high levels. It's been dumped into the cell. Does that make sense? Because again, we need this to set everything back up. So then where's calcium then? Same deal. It's dumped. It's deep into the cell. Calcium is attached. That's why he's like this. All his muscles are contracting because he doesn't have any of this. Yeah. And the worst is when the heart does it, right? Yeah. So Jeff's struggling. So we need to get that going. So again, so basically 5 and 6 are similar, right? So sodium levels are going to be high inside the cell, and then deeper in, calcium levels inside the sarcoplasmic reticulum, well, they're going to be low because the calcium left the sarcoplasmic reticulum, right? You're still going to have it in there, but calcium levels inside the sarcoplasmic reticulum are going to be low because it's all outside of the sarcoplasmic reticulum. How do we get it back in? Again, we need acetylcholinesterase to stop acetylcholine from flowing in. All right, so how do these altered calcium levels affect the positioning? So basically, again, it's decreased inside of here. That means it's increased out here. So I'm going to have excess contraction. I'm going to have excess exposure of the binding sites because of all that calcium. So the position is going to be the actin and myosin are going to be connected and stay connected. So what needs to happen to Jeff's postsynaptic membrane to remedy his paralysis? Well, we need to get acetylcholine off of there. Acetylcholine's on that postsynapse. Acetylcholine's sitting on that postsynapse. That's not allowing sodium to get back out. So then he just stays contracted. So Jeff needs acetylcholinesterase, right? Give him a shot of that, and then that'll go in and tell those acetylcholines, hey, get back into the presynapse. So then they go into the presynapse, and now calcium can go back to the sarcoplasmic reticulum. Now sodium can go back into the outside of the cell, the extracellular space. Okay, and then we get Sandy. She's kind of got the opposite deal going. She hasn't been feeling like herself, quite tired, droopy eyelids. So these are symptoms. So you would be writing these down or typing these out. Droopy eyelids, weakness in her arms and legs, difficulty talking. She hasn't been looking happy at work. So she's got kind of that, like, RBF, they call it. Hey, one day at lunch, she started choking on food. So she's having trouble even chewing and swallowing and eating, let alone talking, let alone moving. So it sounds to me like she's got muscle problems, right? Lack of muscle strength. All right. So Heimlich is convinced it's not time to go to the doctor. Yeah, she probably could have gone to the doctor a while ago. All right, so we look at her levels, and for the most part, they all look pretty good. There are a few that are a little bit low, and a few that are a little bit high, but for the most part, everything is within range until we get to the very bottom, where we see antibodies for ACH receptors are present. So antibodies for ACH receptors. So what that's telling us is that postsynapse, the ACH receptor sites on the muscle, are unavailable. So acetylcholine gets released, but it doesn't go to the receptor sites. So those are the symptoms, antibodies for number two. How would antibodies again affect the neuromuscular junction? So they're going to inhibit ACH from attaching to those binding sites. So then we can't ever tap the domino. So it just stays put. See you, Ricky.
Are you gonna be here after class? I'm sorry.
Maybe I might. I've got some things I got to do. I have another class 11:40.
I'll just zoom.
Okay, cool.
Thank you.
All right, talk to you next week. All right, acetylcholine and sodium. So again, if I can't tap that domino, then sodium levels are going to remain where they are. So how would antibodies affect the influx of sodium? Oh, they would inhibit it. There's not going to be an influx of sodium. Basically, it's the opposite of Jeff. How would antibodies affect the levels of calcium inside the sarcoplasmic reticulum? Well, they wouldn't really affect them much. They're just going to be staying where you're going to have the levels inside the sarcoplasmic reticulum are going to stay what they are. Normally, the contraction they go down, right? Because the calcium leaves the sarcoplasmic reticulum. But in this situation, the calcium levels are just going to be regular, normal, even high in the sarcoplasmic reticulum. And therefore, we're not going to have actin and myosin connecting because you need calcium to leave the sarcoplasmic reticulum and attach to the binding site in order to get anything to happen. All right, does that make sense? So then we have to figure out what disorder, and you don't need to do the table below, by the way. I think it says that over here. Yeah, you do not need to do the table. All right, so what what problems does Sandy have? What disorder does she have? Again, you'd have to Google antibodies against acetylcholine. Antibodies for ACH receptors. If I copy that, that's the primary cause of myasthenia gravis. So she's got an yeah, if you have antibodies that are working against you, not for you, then that's an autoimmune disease. So she's got myasthenia gravis. Okay, so destroys or blocks the neuromuscular junction. Diagnosis, the presence of these antibodies in the blood. Treatment, management involves immunosuppressants, thymectomy, so thymus gland, or plasma exchange. The thymus is your immune. That's where all of your immune, your T cells. So yeah, sometimes they'll zap that sucker if you've got a hyperactive immune system. Hmm. We're seeing more and more of this, actually, myasthenia gravis. So anyway, that's your case study 4 and your muscle physiology. Right. Have questions, comments? All right. So Nick's in there. All right. Well, I'm going to stop recording.